The role of ovarian aging and female reproductive aging in particular has really started to finally be recognized as potentially a place to look for some of the earliest events that we can see functional declines that happen during normal aging.
This is literally like the earliest decline. It has huge impact on brain health, cardiovascular health. When women have breast cancer or at risk of breast cancer, one of the first-line therapies is to put them on aromatase inhibitors. It actually gives you accelerated aging with cognitive problems, joint problems, bone density problems. It's very clear that beta estradiol has anti-aging effects in women. And actually, late menopause, best predictor of longevity in women.
My name is Matt Kaeberlein, and welcome to the Optispan YouTube channel. Hey everyone, welcome back to the Optispan podcast. I'm your host, Matt Kaeberlein. Today's episode is an interview with Dr. Berenice Benayoun from the USC Davis School of Gerontology. I was fortunate enough to be able to catch up with Berenice at the 2024 American Aging Association annual meeting, where she was being recognized with the Vincent Cristofalo Rising Star in Aging Award, a very well-deserved award for her many contributions to the field of aging biology. Berenice actually gave two talks at the meeting. One was about the work in her lab on the African turquoise killifish. The other had to do with macrophage aging in mice. We talk about both of these topics. We also do a pretty deep dive into her work on sex differences in aging. It's a wide-ranging conversation. I'm sure you're going to enjoy it. So without further ado, Dr. Berenice Benayoun. All right. Hey, everyone. Welcome to the Optispan podcast again, coming to you live from the 2024 American Aging Association annual meeting. And I am thrilled that I was able to talk Berenice Benayoun into joining me for a conversation. So Berenice is now an associate professor.
I am for since March.
It's awesome. So recently minted tenured professor at the USC Davis School of Gerontology. And Berenice is this year's keynote speaker for the Nathan Schock Center Symposium for the Vincent Cristofalo Rising Star in Aging Research Award. So congratulations on the award. And you also gave a second talk today. So your talk yesterday, which actually you're going to have to fill me in on. So that's one of the things I want to hear, because I unfortunately wasn't able to hear your talk yesterday. So I want to talk about that, was about sex differences in mice, right?
Exactly.
And then today you gave a second talk at the AGE meeting about the work you're doing on killifish. And so we should also talk about that. Now, you did your postdoc with Anne Bernay.
I did.
And I should know this, but I don't. Were you interested in aging before you went to Anne's lab?
Yeah. So actually, that's a long and funny story. I don't know if you'll want it on the podcast, but basically, I always knew I wanted to be a biologist as soon as I understood what biology was. I didn't know what part of biology I was interested in. And so I took this route, in the French education system, like a general genetics biology degree. And summer of the end of my undergrad, I knew that I wanted to have a research experience in the US, because as good as research is in France, and I think we have amazing people there, the funds to do research are laughable. And so I wanted to experience, a US lab with the means that it has. And so I talked to one of my professors, Olivier Bensaud, and he told me, I have this pal, Rick Morimoto. He's at Northwestern in Chicago. You should email him and tell him, that I'm the person, sending you his way, and see what happens. And Rick was gracious enough to host me for a summer. And I didn't know anything about anything aging.
And Rick, of course, is extremely famous, one of the leading people in proteostasis and aging. And
Exactly. So I started to get exposed to that. And he sort of sicked me on his German postdoc, Klaus Richter, who's now a PI back in Germany, who was doing this big genome-wide RNAi screen in C. elegans for modifiers of the, chaperone response. And, he was probably like, what am I going to do with her for 3 months? So he ordered a couple of worm strains from the CGC, including a DAF16 reporter. and told me like, look, if my RNAi screens do anything, my RNAi screen hits do anything in those. And turns out like I got a cool phenotype with the DAF16. And so I started, reading on it, writing my little summer internship report and discovering all of this realm of aging being regulated. And that sort of drew me in, the idea that, it's not just decay. There's actually a method to it, a regulation, and that maybe we can do something about it. And that's actually the second funny part, is because when I was doing that, DAF16, the worm FOXO, I became familiar with Anne's work.
Right.
And I became a groupie, basically. I was like, oh my God, this is like awesome, especially because she's an alumna of the same school I went to in France.
Okay.
And so I was like, I want to be her when I grow up, basically. And so...
Does Anne know that?
She does.
Okay, good.
And so, the next year, so I was first year of master's, I had a 6-month internship that I could do abroad. I reached out to her. Her lab was full, but she was like, if you ever decide to like do this again, contact me. And at the time, I thought I would go back to France eventually. So I was like, well, I'm going to still do my PhD in France, but I'll want a postdoc. And so basically, everything I did during my PhD was with the idea of I want to have an attractive CV to join Anne's lab to do my postdoc. And so my PhD advisor jokingly always said, you only care about Anne Brunet.
And it worked, right?
It worked.
You were successful. So that's great.
So I was interested in aging. But so my PhD lab was working on FOXL2, which is another fork-head transcription factor, which basically looked like it was involved potentially in regulating ovarian aging.
Okay.
But when I joined my PhD advisor's lab, they were looking more at the developmental effects of that gene. And basically, we had this funny thing where he was like, okay, I'm going to give you 6 months. When I joined his lab, he's like, I'm going to give you 6 months to give me some inkling that there's some relevance to aging. And if you give me something, then you can work on that. Otherwise, you'll work on development like everyone else in the lab. And I got the inkling, so I was allowed to do that and get into that. But that basically, even though it's sort of a secondary idea, right? The ovarian aging came because I wanted to have some aging aspect in that lab.
Yeah.
Then now it's really, with everything I learned and realizing the impact of ovarian aging on somatic aging,
right?
That's sort of then combining with what I did with Anne. I was like, I want to bring those two things together. Right.
And I mean, I think you can probably comment on this. It's my perception that the role of ovarian aging and female reproductive aging in particular, just in the last few years, has really started to finally be recognized as not only really important, but also potentially a place to look for some of the earliest events that we can see functional declines that happen during normal aging. And so you were very early to this.
No, definitely. And I'm so glad that people are starting to be cognizant of that. Because like, again, during my PhD, when I was telling people I was working on that, like the response you get is, but who cares if women can have kids when they're 70? And it's like, that's not the point. This is literally like the earliest decline. It has huge impact on brain health, cardiovascular health.
Right.
And there's nothing being done. So I'm so glad that there's now work being done and more people taking this into account, because I think that has the potential to be transformative for women's health during aging.
Right. And when you think about, I mean, there's a lot of angst in the field about, how do we do clinical trials for aging? Yeah. Well, that's a perfect place, right? Where you actually have an endpoint in a population where you know what to measure, and, you could actually show efficacy of an intervention. So I think
at
for many reasons.
Short, timeline. Yeah.
Right. Okay. Very cool. So can you just give us like a brief version of the talk you gave for the Shock Center?
So for yesterday's session, I talked about a project that my lab's been working on pretty much day one, which was this complete shift actually from the postdoc, because I wanted to, again, look at sex differences, but also looking in a population that would be interested, which was in immune cells. And we decided to focus on macrophages. And basically, when we started this project, we were very agnostic as to what we were going to see. I expected aging to have the bigger impact. But what we saw, and that I reported yesterday, was that at the molecular level, when we measure everything we can measure in those cells, RNA, chromatin, the biggest difference is always whether the animal was female or male, not whether they're young and old. There's definitely an aging effect, but that's not the biggest effect. Now, what's even more interesting and different than previous studies, even of sex dimorphism, is that not only they're different to begin with, but they also age along different trajectories, right? So the changes that occur with aging in the female macrophages are different than the changes that occur with age in the male macrophages.
This is at the level of transcription
and chromatin.
Okay.
And so using that data, and that's like a strategy we like to use a lot in the lab, is generate the big data, the big unbiased data, and then try to see what it tells us about function and then potentially fix it, right? Once we figure out what drives the problems. And so when we looked at functions based on that transcriptomics and epigenomics that were specifically changed in one sex and not the other, the biggest hit is basically phagocytosis, being all like phagocytosis related genes being downregulated in the females with age, but not the males. And so we went and did functional validation of that. And actually the females have a specific defect in phagocytosis with age that the males do not have.
But both males and females show functional deficits.
In other ways, but not for phagocytosis.
Okay.
And we also then observed a metabolic defect that's also specific to the females, where normally you want for macrophages not to be super inflammatory, which isn't good in the long run.
Sure.
You want them to be more respiration biased and less glycolysis biased, because when they're more glycolytic, they will be more inflammatory, they will produce more ROS, and so they will...
So do females have a hyperinflammatory phenotype with age?
Yes. Much stronger than the males. The males have an increased inflammation, but much more subtle than what's going on.
Is that true in humans as well as mice, or is that known?
So it's, I think actually in humans, what is known is that, again, basally the males start out more inflammatory. So that's always the case. So it means the increase with age is not as violent, basically.
Yeah, interesting.
So so we saw that, and then basically a big strategy that my lab uses when we find those sex differences is to try to figure out, okay, what drives it? And, you always have the potential chromosomal component being XX versus being XY, or the potential hormonal component.
Right.
But because here it was not like a basal difference, but a age-induced difference, we thought it might be linked to the decrease in circulating estrogens. And mice don't get menopause. They get estropause, and I know this might seem like I'm doing semantics, but it's actually a 30-fold decrease in estrogen circulation compared to a 300-fold in humans. So
just for the non sort of mouse aficionados, is that the primary difference? Like, what is the difference between estropause and menopause?
So menopause needs to lead to basically undetectable estrogen.
Okay.
You need to get to a level where there's nothing left.
Okay.
Whereas for the mice, they get to a level that's very low but sustained. And because of that, there's still a lot of estrogen receptor signaling that can happen.
But they do become infertile. They do stop cycling.
Become infertile. They stop cycling, but they don't get to zero oocyte in the ovary.
Got it.
Like
and that happens at
do.
What, like about 12 months?
10 to 12
mouse?
months old is when estropause happens. But that's,
in terms of like the total lifespan of the animal, that's actually earlier.
It's earlier than humans.
Than what we see in, Yeah.
But because they keep some estrogen signaling, it's not going to be as bad for like bone density and everything like it is in humans.
Yeah, okay, that's important,
But so we thought maybe that can explain things. And so to make a long story short, we tested what happens if we forcibly remove estrogens in a young animal. And, obviously that's the sledgehammer approach, just remove the ovaries and see what happens. And we're able to phenocopy the loss of phagocytosis, but there's no effect on the metabolism. And then we also did a late life supplementation with estrogen, so sort of estrogen replacement therapy, but very late life, so just for a month, which would be the equivalent of giving estrogen to like a 70-year-old woman, right? So has not seen estrogen at that level for a very long time. And so we have partial transcriptional rescue of aging by doing that. But functionally, the effects are very much dim. And so what I was explaining that we're really excited to have funding to do now is actually long-term supplementation starting around periosteropause.
Right.
Because there's data in humans with the Women's Health Initiative trial, actually the one that made everyone afraid of estrogens. If you know that the trial was stopped because they were like, oh my God, it's giving women cancer.
Right, breast cancer, right?
But breast and ovarian cancer. Yeah. But when people went back to the data years after the fact and stratified by how long women had been menopaused before they were put on the hormones, all of the cancers were in women who'd been menopaused for over 15 years, basically. And all the women who'd been menopaused for less than 5 years had all the benefits and none of the problems. And so because estrogen signaling has a lot of feedbacks on the expression of the receptors and everything, it's what's believed to be the case now in the field is that the longer you go without estrogen, the more poorly you're going to know to respond to them. And so if you start supplementing before you lost the memory of how to respond to them, you'll respond better.
Yeah, that's interesting. So this is a little bit off topic, and maybe you don't know the answer, but is there any evidence for the same thing with testosterone in men? In other words, men that may have had very, very low testosterone for many decades or a couple of decades.
So I don't think there's feedback signaling on the androgen receptor from testosterone.
Okay, interesting.
So my
speculation,
my wild guess would be that they wouldn't lose the ability to respond properly.
Okay, interesting.
There's also only one testosterone receptor, and there's at least 3 estrogen
which
receptors.
Complicates things a lot.
And the interplay, because the estrogen receptor alpha and beta can actually do heterodimers, which don't have the same targets that the homodimers. And then you have the whole estrogen receptor X, where when you remove all the estrogen receptors we know about, so estrogen receptor alpha, beta, and the G protein coupled one, they're still signaling by estrogen. So we know there's a receptor that we don't know about, basically. So people call it receptor X.
Which is super interesting. I think very few people watching this would realize that there's this estrogen receptor that we just don't even know what it is, where it is, what gene it is.
That makes it difficult to understand what
there's a lot of biology that we don't understand.
But so we think the long term will likely be amplifying the results we saw short term and will give us better data. But because none of those, so this seems to be explaining the phagocytosis defect, but it's not explaining the metabolism at all.
Mm-hmm.
So we went and did sort of a deep dive into our omic data and, doing a lot of mumbo jumbo that I'm sure your viewers are not interested in. We were able to identify a few potential candidates that would mediate that non-estrogen mediated female-specific changes in transcription. And we zoomed in on this transcription factor called IRF2 that seems to be sufficient to explain the metabolism changes that are specific to the females with age.
But this is not regulated by estrogen.
So it's independent of estrogen. And so there's 2 ways this could happen, and we have some experiments plans to try to figure it out. It could be potentially downstream of, being XX versus XY.
Mm-hmm. Right.
Or it could be a niche interaction effect, right? Because the macrophages have the estrogen receptors, but the cells around them do too. And so differential, signaling could have those effects as well.
Right.
What we do know is even though the IRF2 is only downregulated in the female macrophages with age, and it's already low in the males to begin with, when we look at our estrogen supplemented female mice or our OVX mice, IRF2 does not change at all in there. So that suggests that the way it is regulated is at least not directly through those hormones.
Right.
Those hormonal changes.
Right. Very cool. So a couple of questions that may or may not be at all well. So one question is, presumably this is, you looked at chromatin structure, there's epigenetic regulation going on. Are the epigenetic clocks sex specific? So
we haven't looked at DNA methylation specifically in that model, but the short answer based on the literature is they are. And what's really cool is in humans, actually going back to menopause, before menopause, the clocks tick slower in females.
Yeah, I know that data.
Males.
Yeah.
After they accelerate.
Right.
And from another project entirely, we've done some DNA methylation analysis on aging mice, and they're not wild type. They're like this weird sex reversal mouse model that my lab's developing, but it does look like the females are taking slower in mice too.
So you can build, so people don't typically use a sex-specific clock though in a lot of the studies, right? You can build a clock that works for both.
It works for both, but it looks like it ticks slower in females.
Okay. And just because we keep going off on a little tangents that pop into my head, what do you think about this study that reported that the epigenetic clocks accelerate during pregnancy, and then at least some women revert post-pregnancy? What are your thoughts on
that? As someone who just went through pregnancy and childbirth, I believe it. I actually believe that brings us to another question, right? I keep telling people you need to look at females and males, but you have this idea like all of the animals we use in the lab are virgins, sex-segregated virgins.
Yeah, right.
And so that's not super representative of humans either. And so one of the things we've started to do, we're very early, is to try to compare actually the aging profiles in retired breeders. And obviously those have been bred to death.
Right. So it's like kind of the opposite extreme.
But compare how they're aging to the virgins.
Yeah,
We start
that's
with
interesting.
The extreme.
Yeah. And that's actually a good experiment because, that would be really interesting to know if their epigenetic clock has been accelerated by being breeders or not. I don't know if I should say the results here or not. It's up to you. If you want it on the internet, then say it. Otherwise, it'll be a mystery till the paper comes out.
We have preliminary data showing that the at least way after they've stopped breeding, they're probably rejuvenated.
Okay.
Which would make partial sense with epidemiological data in humans, because we know that having had kids increases the expected lifespan of women.
Let me make sure I understand what you said, though. So you said they're partially rejuvenated. Do you mean that the breeders
their epigenetic age at the same chronological age, at like 13, 14 months of age, they are 3 to 4 months younger than the virgin counterparts?
Oh, that's interesting. So, but what you don't know is whether in mice a given pregnancy event accelerates it.
So but we didn't do it at that.
Still, that's really interesting. Oh, very cool. Okay, so I'm excited to see where that goes. That's pretty neat.
I'm excited too. I want to see that. Where like the new data we're gonna get goes there.
Yeah. So then the other thought that pops into my head is, do you have any thoughts on the sex-specific benefits of 17-alpha estradiol in the interventions testing program? And part of why I'm asking is because you may be familiar with this. There was I think it's just a preprint still. I don't think it's been published, from the guys at Epiturna, where they looked at prescription drug use in the UK, and they found like 16 things that were associated with lower all-cause mortality, and like 6 of them were estrogens, right? Which is only in women in that case. And so I don't have a good even idea for what might be causing these male-specific effects in mice, but apparently some benefits in women from estrogen replacement therapy.
So, like the first thing is really how similar the signaling of the alpha and the beta forms are going to be. I think the male-specific effects of alpha are probably because the way it signals is slightly different. Even if it uses the same receptors, it's not going to have the exact same affinity with the 3 receptors, which means the combined output is going to be slightly different. Now,
do we know in what way though?
No, like Mike Stout has done some really cool work showing that it does go through ER alpha in great part, which people didn't believe before he did that. So that was like really cool to like prove like, no, you remove estrogen receptor alpha, most of the benefits of 17 alpha estradiol are gone. Mm-hmm. Now, what's really cool, and I think that was Rich Miller's group who did that, is you would expect if the only thing it's doing is giving estrogenic signaling to males. And the reason it doesn't work in females is because they already have enough internally. Well, I believe he showed that when you ovarectomize the female mice and you give them the 17 alpha, it actually still doesn't work.
That's right. Yep.
And so, but even though people tell you 17 alpha is non-feminizing, I can tell you from experiments we've done in the lab in collaboration with Christian Pike, you have a uterotrophic effect. So it means that there is some feminizing effects of 17 alpha. And they might not be as strong as if it was the beta, but they're there. So why does it work in one and not the other? My guess is because a lot of the benefits of the of 17 alpha seem to be, hypothalamic. And so it is possible that the way the response is happening in the brain, and we know the brain can be very different in the way it responds to a lot of stimuli and hormones in females and males. My guess is it's going to be that. Now, unpublished, again, but I'm in the collaboration with Christian Pike, we've been giving 17 alpha to female and male mice to see what happens. And even though there's absolutely zero benefits in terms of cognition, we do see metabolic improvements in the females.
In brain?
No, like peripheral metabolic.
Okay.
Just nothing good in the brain for the females.
Interesting. But you do see improvements in the males?
Yes.
Okay.
That's pre-printed.
Okay. And is this in the Het3 or is this in
So those
C57
are in
Black
actually
6?
ApoE humanized mice.
Okay. So this is a cognitive decline dementia.
But they're not like Alzheimer's. We just humanized ApoE, but they're otherwise wild type.
Do they develop cognitive decline differently?
They do develop. The E4s are more challenged than the E3s.
Got it.
But the E3s are basically
Pretty normal.
Come like the same as the wild type.
Okay.
Yeah.
But is that on I'm asking because I don't know.
Those are a Black 6 background.
Yeah, I was going to say, I don't know if anybody's done the 17 alpha estradiol longevity studies outside of Het3. I think all of those have been done in the ITP, but I could be missing something.
I think Mike Stott has some data on the Black 6, but I'm not sure.
Okay.
Yeah.
We'll have to look that up and put it in the show notes.
But yeah, so like the short answer is it's unclear why, but we know that supplementing beta in females is positive. So that's why I think there's like
positive
some of that
for what?
Metabolism, brain, everything.
But nobody's done a longevity study.
Nobody's done the longevity directly. What people have done longevity for that I think is sort of a nice substitute is the ovary transplant. Do you know that study?
Yes, I think so.
So like this really cool study where they took middle-aged mice and did a transplant of young ovaries. They didn't even remove the old ones. They just added young ones.
Right.
And that's enough to increase lifespan.
Right.
And because those ovaries weren't even in situ, I think they were put sub-Q just so that they would, get vascularized. What that's going to do is estrogen with a little bit of progesterone, basically.
Yeah.
And that's enough to
Which is, that's a whole super fascinating area where, you can put young blood in, you can put young ovaries in, you can put young poop in, right? I mean, apparently there's lots of ways that you could take something from a young individual, put it in an older individual and get some benefits, right? Yeah. I'm trying to remember that. I can't remember the absolute magnitude of the effect in that study. And whether the surgery itself depressed the lifespan. That's one of the challenges with, the heterochronic parabiosis literature is kind of like that as well, where the surgery itself has an impact. But still, I believe that there's benefits from doing those kinds of experiments.
Like there's enough data in humans. Again, like when for any reason you're going to have premature loss of ovarian function. Absolutely. That definitely leads to accelerated aging. There's actually some fascinating in a sad way. I don't know if it was with you that was talking about this yesterday, but when women have breast cancer or at risk of breast cancer, one of the first line therapies is to put them on aromatase inhibitors. And basically, even when you're menopaused, your fat tissue has aromatase, and you're going to have some small local production of estrogens, or in the brain too. Actually, for men, there's a lot of production of estrogens in the brain. And people used to think that tiny amount was too little to have any biological impact. But the fact of the matter is, when you put those women who, a lot of them postmenopausal, because that's when the breast cancer will happen,
right,
on aromatase inhibitors, it actually gives you accelerated aging and sort of exacerbated menopause symptoms with cognitive problems, joint problems, bone density problems. And so, and the fact that when you look at the Women's Health Initiative trial cleanly, by looking at those women who were given it really close to menopause, and even now in clinical practice, you have some doctors who have read the data and will prescribe it at least perimenopausally. It's very clear that beta estradiol has anti-aging effects in women, and actually late menopause, best predictor of longevity in women.
Yeah. I mean, I think I agree completely. That's my interpretation as well. And I think, it's funny because people ask, like, what is the best candidate for a drug that might slow aging in people? And that might be it, where the data is actually, I think, the most convincing, at least in women.
No, and really, this is something that riles me up because the Women's Health Initiative trial was designed like ass, let's call it like it is, I think that has lastingly harmed women's health.
No question about it. Yeah. I mean, again, this is, one of the unfortunate aspects of the way sometimes studies are designed, data gets misinterpreted, overinterpreted, and it really can have a pretty huge impact on quality of life for people. And
because the problem is, even if some doctors are now willing to prescribe it, you have women who are going to go on internet to look up the treatment and see all the scary things and refuse to take it. Yeah. Actually, I had this discussion with my mom like 5-6 years ago where, she was interested in what I was working on, and I was telling her about like how estrogens are amazing. And she's like, oh, my gynecologist had prescribed estrogen therapy when I was like around menopause. And I said no, because I was afraid I was going to get cancer. Yeah. And I was like, ah!
Well, especially cancer especially, I think, is something that is just scares people with good reason. And I've had probably 2 or 3 female doctors tell me that they believe every woman
should be on estrogen replacement therapy.
Exactly. Yeah. So I think that, the pendulum is swinging, but there's still a lot of misinformation and fear out there, unfortunately. Yeah. So anyways, it's great that you are doing good work in this space and fantastic mechanistic insights that,
thank you.
Yeah, looking forward to see where that goes. So let's switch gears a little bit and talk about Killifish. So
let's talk about Killifish.
So tell me about Killifish. I mean, tell our audience about Killifish. I know a little bit about Killifish.
No, so they're this really cool fish. So to start with, for people who aren't familiar with the limitations when we're working in aging, animals. On the one hand, you have, C. elegans, flies, super short-lived, super powerful, cheap to use, but they don't recapitulate things that are specific to vertebrate aging. On the other hand, you have mice, zebrafish, great. They have the bones, they have the blood, they have the complex brain, but they live forever. Mice about 3 years, zebrafish 6 years. And so that's not manageable. And so the impetus behind developing the African turquoise killifish is it's literally the shortest lived vertebrate that can be bred in captivity. And so the standard lab strain that we use, the GRZ, live about 4 to 6 months. And that's basically the lifespan of a fly. And so you have invertebrate lifespan with vertebrate biology. So that's the beauty of that model.
So when did you start working with Killifish? Was that in Anne's lab?
That was in Anne's lab. And that's another funny story. And I want to say, I don't know if like trainees watch your podcast, but that's like sort of a very serendipitous want to help a lab mate story. Like I was just doing mammalian work, and Dario Valanzano, who brought the killifish to Anne's lab, was trying to do this genetic association using the different strains that have different lifespan to see if we could find genetic associations in that species. But obviously, even if you have the linkage map, you can't know what gene is next to your marker unless you have a genome. And at the time, they had done like a bunch of Illumina sequencing for the Killifish genome, and they were waiting on a collaborating lab to assemble the genome and annotate it. And you know how it goes. Another lab, that's not their project. It's not their priority. And Dario was getting so frustrated. Like, he was like, you know, this is taking forever. It's driving me nuts. And at the time, I was learning to do a lot of that computational stuff in bioinformatics. And I was like, do you want me to take a stab at it? And so I took a stab. I assembled the genome, and the rest is history. But like, you know, that really came out of I'm seeing my lab mate being super frustrated, and I offered to help.
And so you brought Killifish into your own lab. Now, when did you first start working on Killifish? Did you bring them with you when you started at USC?
So I waited a little bit because I wanted to first like start a lot of the, you know, starting 2 models at once seemed like a lot. So I waited a year into getting at USC because then I needed to also, you know, it's a species nobody at USC was working on.
That's what I was going to say. Are you the only person there? So you had a lot of infrastructure
and also a lot of negotiation with the veterinarians to explain
because they aren't used to it.
Yeah, because they think it's like a zebrafish, and you're like, no, it's like not the same temperature, not the same water quality. So we had to do a lot of negotiating.
Yeah, and that's actually something that probably a lot of people watching aren't going to appreciate. But because it's a vertebrate, you are required to go through most, maybe all of the same animal care approval processes you would if it was a mouse or even a non-human primate.
Yeah.
Whereas C. elegans, you don't have to do any of that stuff. So
nobody cares if you kill
the paperwork is much less of a burden for invertebrate models. So yeah.
No, definitely. So it was about like a year in to USC that I got the system, and we started like getting them going. But that's the beauty, is actually a lot of what we've published. You know, I've been at USC 7 years. I think the majority of the publications we have on the Killifish, despite the fact we started it a year after the mouse.
Yeah, so. Well, yeah, that's the beauty of working with a fast-aging animal, right?
Yes, definitely. So yeah, it's a really cool fish. So it comes from those ephemeral ponds of water in Zimbabwe and Mozambique, where basically you have water for 4 to 6 months during the year, and the rest of the year it dries up. And so they survive the dry season as eggs in the dried mud. But they basically had to evolve what Anne calls life in the fast lane, because they need to, as soon as there's water, go, hatch, reproduce, you know. And so we think part of the reason they age so fast is because they do not spend any resources maintaining their soma.
Right.
And so, you know, relevant to that, we see that they have very poor, like the mutation rates are insane how fast they mutate.
And so what are the key questions you're most interested in asking in Killifish?
So because of their, you know, crazy short lifespan, they also have, so their genome has characteristics that are much closer to the human genome than the mouse genome. And like one feat in particular is the amount of transposons in their genome. So again, for those of you who may not know, transposons are these like selfish genetic elements that basically their entire goal is to just amplify and increase the number of copies in the genome. They don't contribute much. People used to think they were just junk DNA and contributed nothing, but there's actually more and more data suggesting that they do contribute to the host biology. Like the reason we have placenta as placental mammals, it's actually derived from a transposable element protein.
I did not know that.
And there's some data suggesting that long-term memory formation in the brain might be aided by transposons. So they contribute. They're not completely freeloaders. But the human genome is about 50% transposons. The mouse genome is only 35%, so much less.
Which is a huge fraction. I mean, I'm sure people listening to this are going to be like, what the 50%?
And most people ignore it because it's just complicated to look at. The Killifish is, so we're not fully sure because the assembly is not as good quality as the human genome, but it's between 65% and 55% of the genome is transposons. Okay. And so we are in a range where we can actually see if the regulating those transposons and controlling them has impact on aging, because not only there's a huge amount, but they're also active pretty early in life. And so that means that so one of the things we're looking at is what happens. So do they get deregulated with age? And they do, they accumulate.
And I mean, that's one thing that's probably worth saying is, right, over the last maybe decade or so, I think it's become pretty well appreciated that during aging, in maybe all mammals where this has been looked at least, you see a general dysregulation of transposons.
Not just mammals, like flies and worms too.
Yeah. Okay. So maybe across the entire animal kingdom, maybe even more than that. But you see a general dysregulation of transposable elements, a lot more apparent jumping around. And that can have a lot of consequences, right? If you have these random DNA sequences excising and then integrating back in.
So that's one of the ways they can be bad, but they can actually be bad without even reintegrating because they, it's like, talks like a duck, walks like a duck. It looks like a virus.
Yeah.
And so your cell, when you express transposons, all the pattern recognition receptors that have evolved to recognize viruses, they get activated by those transposons. So MDA5, MAVs, all of those get activated. And so even if they don't reintegrate, they're going to sort of turn on all those inflammatory pathways.
Right.
And so, part of the question in the transposon and aging field is their activation just a byproduct of aging, or could it actually drive?
Yeah, no, absolutely. Well, and I mean, it's sort of interesting to think about, right? Because presumably one of the reasons why you get this activation of transposons is because of epigenetic dysregulation. Yeah. But then the jumping around of the transposons could drive epigenetic dysregulation, right? So it's sort of like this vicious cycle and DNA damage and inflammation,
No, and and because of the link with MAVs, which is a detection mechanism for that, there's also metabolic, that can also lead to mitochondria dysfunction, which is also a hallmark of aging.
Right.
And so, that's why we think this fish is actually a great way to test this whole hypothesis of the chicken and the egg in terms of our transposons potentially, the deregulation of transposon potentially causal. So
how are you going to test that? What's the experiment?
So we're doing that in two ways. We are using genetics, looking at known regulators of transposons, see if we can, overexpress them to death to force transposons to calm down, and see how that's going to impact cognitive aging, lifespan, health span. Another thing that we're excited to do, and that I'm hoping my grad student will do, is actually feed anti-reverse transcriptase.
Yeah, that's what I was going to ask, is could you do this pharmacologically?
Yeah. It's like, so again, every model has its pluses and minuses. We can feed drugs, and we've been doing it for another compound. But because it's in the water and you're in a recirculating system, we have to make sure there's no drug staying in the water for too long. So we have to tweezer feed them the food when we're giving them drugs. So it is work intensive. Wow. But it's feasible. It is feasible. We've been working on a specific drug in collaboration with another lab at USC, and we can see lifespan effects, no problem. So we know it is feasible. It's just more work intensive, but that's something that we definitely want to do.
That sounds like a very intensive experiment. And this comes from a guy who did yeast replicative lifespan assays for years of my life. So
I feel like that sounds worse.
I don't know. I've never tried to tweezer feed a killifish, so I can't comment. But
at least you don't need a microscope.
This is true. Yeah. Okay. Well, I don't know. I hope your grad student does the experiment too. I'll just leave it at that. But it'll be interesting to see. I mean, I do think that, this is another area where, even though within the field, I think most of us are aware of this area of research, it hasn't really gotten as much attention outside, and it has a lot of promise. So be interesting to see where it goes.
Yeah, definitely.
Cool. So that's fantastic. Anything we haven't talked about that you're burning to share with the audience here?
I think we covered the big ones. Again, a lot of what my lab is doing is looking at the stuff that everyone else doesn't want to look at. So I don't know what that's actually
not a bad place to be. It's not a bad place to be at all. No, you're doing really cool stuff. So even if nobody else is looking there, it's because they're too dumb to look there. I think you're doing great stuff. So I would not worry about that.
No, thank you. That it's actually been really great to see, people get excited about the things that I'm excited about.
Yeah.
So
Yeah. All right. Well, thank you so much for sitting down, and
Thank you, Matt.
We'll look forward to getting an update maybe at next year's meeting. Are you going to go to
pleasure.
Alaska?
Oh, I'm excited to go to Alaska. I've never been.
Cool.
So yeah.
Awesome. All right. Well, thanks guys for watching the OptiSpan podcast. As always, if you have any questions or comments, please leave them below. If you're not yet a subscriber, hit that button, and we'll see you next time on the OptiSpan podcast.